Soft background music playing. Narrator, welcome to today's lecture, where we will delve into the fascinating world of microscopy techniques in medical microbiology. In this comprehensive script, we will explore various microscopy methods used to study and diagnose microbial pathogens. So, grab your lab coats and get ready for a microscopic adventure. Professor, microscopy is an indispensable tool in medical microbiology, allowing us to visualize and study microorganisms at the cellular and subcellular levels. It enables us to identify pathogens, understand their structure and function, and aid in the diagnosis of infectious diseases. There are several types of microscopes utilized in this field, each with its unique capabilities. Professor, light microscopes, the most common type, employ optical lenses to magnify and visualize samples. They consist of various components such as the ocular lens, objective lens, condenser, and light source. The ocular lens provides the observer with a magnified view, while the objective lens gathers light and magnifies the sample. The condenser focuses and illuminates the specimen, allowing for clearer visualization. Professor, advanced light microscopy techniques have revolutionized medical microbiology. Techniques like confocal microscopy, phase contrast microscopy, and fluorescence microscopy provide enhanced resolution, 3D imaging, and the ability to visualize specific components within cells. Confocal microscopy, for instance, uses lasers to create optical sections, allowing researchers to examine layers of cells. Professor, electron microscopy takes imaging to another level by using a beam of electrons instead of light. Transmission electron microscopy, TEM, and scanning electron microscopy, SEM, are the two major types used in medical microbiology. TEM provides ultra-high magnification, allowing for detailed visualization of cellular structures, such as bacterial cell walls. SEM, on the other hand, gives a three-dimensional view of the sample surface, enabling us to study surface characteristics of pathogens. Professor, immunofluorescence microscopy employs fluorescently labeled antibodies to detect specific antigens on pathogens. This technique allows us to identify and localize pathogens within patient samples. By visualizing the fluorescent signal, we can assess the presence of specific microbes or antigens in various clinical samples, aiding in the diagnosis of infectious diseases. Professor, digital microscopy combines the power of advanced optics with computerized image analysis. It allows for automated capturing, archiving, and analysis of microscopic images. This integration enhances the speed and accuracy of microbial identification and characterization, facilitating more efficient diagnosis in clinical settings. Professor, microspectroscopy techniques, such as Raman microscopy, enable us to study the molecular composition and structure of pathogens. By illuminating the sample with laser light and analyzing the scattered light, we can gather valuable information about the chemical composition of microbial cells. This aids in the identification and differentiation of various microorganisms. Professor, atomic force microscopy, AFM, provides ultra-high resolution imaging of microbial pathogens. This technique uses a sharp probe to scan the sample surface, detecting atomic or molecular forces. AFM can visualize structural details of individual bacteria and viruses, aiding in pathogen characterization and evaluating the impact of antimicrobial treatments. Professor, cryo-electron microscopy, cryo-EM, has revolutionized our ability to study the three-dimensional structures of microbial pathogens. By rapidly freezing the sample, we can preserve its native state and visualize its structure with high resolution. Cryo-EM has played a crucial role in understanding the architecture of viruses, bacteria, and other infectious agents. Professor, single-molecule microscopy provides insights into the dynamic behaviors of individual molecules within microbial systems. This technique allows us to observe and track interactions between pathogens and host cells, as well as study molecular processes such as gene expression and protein-protein interactions. It helps unravel the intricate mechanisms underlying pathogen-host interactions. Professor, correlative microscopy combines multiple imaging modalities to generate a comprehensive view of a biological sample. By merging data from light microscopy, electron microscopy, and other techniques, we can correlate structural, functional, and molecular information of pathogens. This holistic approach enhances our understanding of complex microbial systems. Professor, advanced microscopy techniques have paved the way for rapid infectious disease diagnosis in clinical microbiology. Innovations such as point-of-care microscopy devices and automated image analysis algorithms have significantly reduced turnaround time and improved diagnostic accuracy. These advancements have a profound impact on patient care and public health. Thank you for joining us on our exploration of microscopy and medical microbiology. These powerful techniques provide vital insights into infectious diseases, enabling scientists and doctors to better understand and fight against them. From traditional to cutting-edge microscopy, these tools hold the key to unraveling the mysteries of pathogens. By advancing microscopy, we pave the way for exciting breakthroughs in medical microbiology.